Welcome back to the deep dive. Today, we are stepping onto a battlefield.
Hmm.
But it's not a historical one, and it's not one you can see on a map. It's the battlefield inside your own body. We are looking at the immune system.
And specifically, we're looking at what happens when that system, which is, it's arguably the most sophisticated defense network in existence, goes wrong.
Exactly. I think most of us grow up with this idea of the immune system as just the good guys.
Well, sure.
You know, the white blood cells are the knights in shining armor fighting off the bacteria and the viruses. But the sources we have today paint a much more complex and, frankly, sometimes terrifying picture. It's not always a hero story, is it?
It is definitely complex. The immune system is a miracle of discrimination. Its entire job is to distinguish self, your own tissues, your own organs from non-self, like a virus or, a splinter. When that works, you stay healthy.
Right.
But this deep dive is based on the byline, when defense loses restraint and turns inward.
When defense loses restraint. That is a powerful image. We aren't talking about just having a weak immune system here, are we?
No, not at all.
We're talking about excess. We're talking about misdirection.
Precisely. We are going to explore what happens when the control mechanisms fail.
Sure.
It's the difference between a disciplined army protecting a city and, a riot that burns the city down.
Now, to guide us through this, we have a stack of material, but we are primarily synthesizing two absolute giants in the field of medical education. We have Robbins and Catron pathologic basis of disease.
The Bible of pathology.
Basically.
And Robbins is essential. It provides the breadth, the clinical integration, and that unifying systems framework. It tells you how the whole machine fits together.
And we are pairing that with Underwood's pathology.
Which is fantastic for clarity. Underwood really grounds disease as a response to injury or stress. It gives us those mechanistic explanations that, really help the concept stick.
I want to be really clear to you, the learner listening right now. This deep dive is your conceptual map. Think of this as a scaffold. We are going to help you understand how to read these textbooks, but we are not replacing them. You still have to open the books.
Absolutely. Reading remains essential. We are here to help you connect the dots so that when you do read the chapters on diseases of the immune system, the information has a place to land in your brain.
So let's preview where we are going. We're going to start with the architecture of defense, the normal stuff.
The foundations.
Then we're going to look at the overshoot hypersensitivity, then the breakdown of tolerance autoimmunity, and finally, the gap in the shield immunodeficiency.
And through all of that, keep one key takeaway in mind. Immune disease is often a loss of regulation, not just a failure of function.
Okay, let's unpack this. Part one, the architecture of defense. Before we can understand how the car crashes, we need to know how it drives.
Right. And to keep it simple, we generally divide the immune system into two massive arms, innate immunity and adaptive immunity.
Innate and adaptive. Let's start with innate. This is the first line of defense, right? The front line.
It is. Robbins describes innate immunity as always present, ready to react. It doesn't need to learn who the enemy is. It just knows what an enemy looks like, in a general sense.
Underwood uses a great concept here, focusing on barriers.
Yes. The most obvious innate immunity is mechanical, your skin. The mucociliary lining in your lungs that sweeps out dust and bugs.
The escalator.
The escalator, yeah. The acid in your stomach. If those barriers fail, you get infection. Underwood gives the example of cystic fibrosis, where that mucociliary escalator breaks down, and the result is recurrent, devastating infection.
So that's the wall around the castle. But what happens if something gets over the wall?
Then you meet the cellular arm of innate immunity, the guards. Phagocytes like neutrophils and macrophages.
The eaters?
The eaters, yeah.
Yeah.
You got natural killer cells, and the complement system, which is this cascade of proteins that punches holes in bacteria.
I love the way Robbins explains how these cells know what to attack, because they don't have a specific wanted poster for every single flea virus, do they?
No, they don't. They recognize patterns. And this is a really crucial concept. They use what are called pattern recognition receptors, or PRRs.
PRs.
The most famous of these are the toll-like receptors, or TLRs.
Okay, pattern recognition sounds nice and all, but what are they actually looking for? Does a bacteria have a specific shape they recognize?
It's more primal than shape. They are scanning for molecular signatures that simply do not exist in humans. We call them PAMPs, pathogen-associated molecular patterns.
A PAMP. Can you give me a tangible example? What does that actually mean?
Sure. Think about lipopolysaccharide, or LPS. It's a component of the cell wall in certain bacteria, gram-negatives.
Okay.
Humans do not make LPS. We don't have the genes for it. It's completely foreign. So if a toll-like receptor on a macrophage feels the chemical structure of LPS, it pulls the trigger.
It doesn't even care which bacteria it is.
Doesn't matter. The chemical signature alone is the declaration of war. It's a fundamental sign of non-self.
So it's like a barcode scanner looking for a barcode that says not made on Earth, or at least not made in this body.
Exactly. Or they look for damage-associated molecular patterns.
Damage. So this is our own stuff.
Yes, this is our own stuff in the wrong place. These are things released when our own cells are injured or die messily. If a cell bursts open and spills its DNA or its ATP into the extracellular space, the innate system sees that and says, whoa, that shouldn't be here. Something is wrong.
Cellular guts on the outside.
Cellular guts on the outside. It's a sign of injury, and it kicks off inflammation.
I like the analogy of the castle guard. The guard doesn't know the specific name of the invader, but he knows that anyone not wearing our uniform is a threat.
Exactly. And once they spot that threat, they attack immediately. It's fast, but it's not specific. It's a blunt instrument.
Which brings us to the second arm, adaptive immunity. Robbins calls this specific, powerful, slower.
The adaptive system is the specialists, the special forces. These are the lymphocytes, T cells, and B cells. They don't just attack invaders. They adapt to the specific intruder.
And this is where we get into humoral versus cell-mediated immunity.
Right. Humoral immunity is mediated by B cells. When they get activated, they turn into plasma cells, become these little factories for antibodies, IgG, IgM, IgA, IgE.
So they're churning out these proteins.
Billions of them. And these antibodies flood the blood and fluids to neutralize microbes outside the cells.
So B cells are like the archers on the wall shooting arrows, the antibodies, at the enemy down in the field.
That's a good way to picture it.
Yeah.
Then you have cell-mediated immunity, which is the T cells. And you have two main types of T cells you need to know, CD4 plus helper T cells and CD8 plus cytotoxic T cells.
The CD4s are the generals, right? The quarterbacks.
Yes. They direct the war. They don't kill much themselves, but they secrete cytokines, which are chemical messages that tell everyone else what to do. They activate the macrophages, they help the B cells, they rally the troops.
And the CD8s?
The CD8s are the assassins. They're the hand-to-hand combat specialists. They find host cells that have already been infected, say, by a virus, and they kill that cell to stop the spread. They're taking out the factories.
And the defining feature of this whole adaptive system is memory.
Correct. The first time they meet a pathogen, it takes time to ramp up. A week, maybe 2, but the second time, the response is faster and stronger, and often you don't even know you were exposed. That's why vaccines work.
Yeah.
You're just generating immunologic memory.
So we have the innate guards and the adaptive specialists. But they aren't working in silos, are they? It's not like one team clocks out and the other clocks in.
No, and this is the synthesis point. This is critical. Innate immunity triggers adaptive immunity. There's a cell called a dendritic cell. Think of it as a sentinel or a scout.
Okay.
It lives in the tissues. It captures an antigen out in the periphery, runs to the nearest lymph node, and presents it to the T cells.
You said the dendritic cell runs to the lymph node and presents the antigen. I'm picturing a waiter with a tray. Is it that simple?
Not quite.
Yeah.
And this is where Robbins gets really technical, but it's crucial. The dendritic cell can't just hold up the virus part and say, look what I found. It has to process it, chop it into little peptides, and serve it on a specific plate called an MHC molecule, major histocompatibility complex.
MHC.
And here is the kicker. The T cell won't eat off any other plate. It has to be your MHC.
Wait, so it's a personalized plate. My MHC is different from your MHC.
Yes. This is called MHC restriction. It's a profound security check. The T cell receptors are designed during their development to only recognize a combination of your specific MHC molecule plus the foreign peptide. If the antigen is just floating around by itself, the T cell ignores it.
Wow.
It ensures that the T cell is reacting to something threatening your tissues, presented by your cells.
That's incredibly sophisticated. It prevents them from firing at random trash in the bloodstream.
Exactly. And it also requires what's called co-stimulation. The dendritic cell has to give a second signal, a danger signal, at the same time. It's a two-key system to launch the missile.
Which brings us to the key concept for pathology. Normal immunity is defined by balance, speed, and resolution.
Resolution, exactly. The system turns on, it fights, and then, crucially, it turns off. Pathology starts when the off switch fails or when the target is wrong.
Which leads us perfectly into part 2, hypersensitivity, the overshoot.
Yes, hypersensitivity. Robbins defines this as an excessive or harmful reaction to an antigen, an injurious immune reaction.
I think it's interesting that the definition emphasizes that the reaction itself is injurious.
Right.
The problem isn't the weapon, the antibodies, or the T cells are doing what they're designed to do. The problem is the control and the target.
Exactly. The gun is working fine, it's just being fired in a crowded room. Now, classically, we structured this around the 4 types, the Gell and Coombs classification.
Yeah.
But let's focus on the mechanism rather than just memorizing a list.
Okay, type I, immediate hypersensitivity. This is allergy.
This is the rapid reaction. You get exposed to pollen, and minutes later, your eyes are watering. The key players are Th2 cells, IgE antibodies, and mast cells.
Let's slow this down and look at the mechanics, because Robbins makes a big deal about the sequence here. I breathe in the pollen ragweed, let's say. What is the very first domino to fall?
Well, first, that pollen has to get through the lining of your nose.
Yeah.
Once it's in the tissue, an antigen-presenting cell, probably a dendritic cell, grabs it.
Okay.
Now, in a non-allergic person, the immune system might ignore it or just produce a mild IgG response. But in an allergic person, someone with atopy, the response is hijacked by a specific type of T cell, the Th2 cell.
The Th2, this is the one associated with parasites, like worms.
Correctly. And that's the evolutionary irony. The entire system is beautifully designed to fight intestinal worms, and now it's fighting grass pollen. The Th2 cells secrete specific cytokines, primarily IL-4 and IL-13.
We talk about cytokines a lot, but what are IL-4 and IL-13 actually saying to the army? What are the orders?
They are very specific instructions to the T cells. They scream, switch production, stop making the default IgM and start making IgE. This is called class switching.
Okay, so now the B cells are pumping out IgE specific for ragweed pollen, but IgE doesn't kill anything on its own, does it?
No. IgE is just a key looking for a lock. It floats through the blood until it finds a mast cell. Mast cells are everywhere, under your skin, in your lungs, near your blood vessels. And they're studded with high-affinity receptors, specifically for the tail of the IgE molecule.
So the IgE sticks to the outside of the mast cell with the antigen-binding part facing outward.
Like spikes on a naval mine. And this is the critical part. This is the sensitization phase. You don't feel a thing. You can walk through a field of ragweed and you won't sneeze once. But internally, your mast cells are being armed. They are coating themselves in these antibodies.
They're waiting.
Waiting for the second exposure.
Right.
The next week, you inhale that pollen again, the antigen lands on the mast cell and physically bridges 2 of those IgE antibodies together. It cross-links them.
And that physical connection is the trigger, that little tug.
That connection sends a signal inside the mast cell that opens calcium channels. Calcium rushes in and the cell undergoes explosive degranulation. It just vomits out these vesicles full of preformed mediators.
Preformed meaning they were already sitting there packed and ready to go.
Yes, primary mediators. The big one is histamine.
And histamine is what makes you miserable.
Histamine is what makes you miserable. It binds to receptors on blood vessels, causing them to dilate. That's the redness.
Yeah.
It makes them leaky. That's the swelling or the running nose. It stimulates nerve endings. That's the itching. All of this happens in minutes.
But that's not the whole story. Robbins differentiates this from the secondary mediators, though, the stuff that comes later.
Yes. This is the late phase reaction, which happens hours later. The mast cell doesn't just dump its trash and die. It starts a factory. It begins synthesizing lipids, prostaglandins, and leukotrienes.
Leukotrienes. Those are the target of drugs that's singular, right? For asthma.
Exactly. Leukotrienes are incredibly potent. They cause prolonged, smooth muscle contraction, which in the lungs means bronchospasm, asthma. They are far more powerful and sustained than histamine at causing that constriction.
And they also act as a beacon.
A huge beacon. They, along with other cytokines, call in other inflammatory cells, especially eosinophils. This is what causes the chronic inflammation and tissue damage you see in things like chronic asthma.
So type A is this massive, rapid orchestration involving Th2, IgE, and mast cells. Let's move to type 2, antibody-mediated.
Okay, in type 2, the antibodies, usually IgG or IgM, are attacking antigens specifically on cell surfaces or embedded in tissues. It's a direct attack.
And there are 3 distinct ways this causes injury. It's not just one mechanism.
No. The first way is opsonization and phagocytosis. The antibody coats the cell, say, a red blood cell, in an incompatible transfusion and signals macrophages to eat it.
It tags it.
It tags it for destruction. The antibody acts like a condiment. It makes the cell tasty to the phagocyte. This is what happens in autoimmune hemolytic anemia.
The cell is tagged for deletion and then eaten. Simple.
Exactly. The second mechanism is inflammation. The antibody binds to a tissue like the basement membrane in the kidney or lung, and it doesn't just sit there. It activates complement.
That protein cascade again.
Right. The complement cascade generates fragments that are powerful chemoattractants for neutrophils. The neutrophils rush in, release their enzymes and reactive oxygen species, and just chew up the tissue. That's Goodpasture syndrome.
So in the first one, the cell is eaten. In the second one, the tissue is inflamed. But the third one is fascinating because it doesn't necessarily destroy the cell. Cellular dysfunction.
This is so elegant and
Mhm.
so terrible. The antibody binds to a receptor on a cell and just messes with its function. In Graves' disease, an antibody binds to the TSH receptor on the thyroid gland.
The receptor for thyroid-stimulating hormone.
Right. But instead of destroying it, it mimics the hormone. It turns the receptor on. It screams, "Make thyroid hormone," constantly, even when there's no TSH around.
So you get hyperthyroidism. The cell is working too hard because of the antibody.
Exactly. Or take the opposite, myasthenia gravis. The antibody binds to the acetylcholine receptor on hustle cells. But here it blocks the signal. The nerve fires, releases acetylcholine, but the antibody is in the way. The muscle can't hear the signal.
The cell isn't dead.
The cell isn't dead, it just can't work, leading to profound muscle weakness.
Okay, so type 2 is direct targeting. Type 3 is immune complex mediated. I always visualize this as a plumbing problem.
That's a very accurate visualization. In type 3, the antigen and the antibody meet in the circulation in the blood and form these clumps or complexes.
And normally the body clears these out.
Normally, yes. Phagocytes in the spleen and liver are constantly filtering the blood and cleaning them up. But in these diseases, there is often an antigen excess. Just too many complexes form at once. They overwhelm the cleanup crew.
So they don't get cleared.
They don't get cleared. They circulate, and then they get stuck. They tend to deposit in small vessels, especially in places with high-pressure filtration, like the glomeruli of the kidneys or in the joints.
And Robbins makes a key distinction here. Unlike type 2, where the antibody is targeting the kidney tissue specifically, in type 3, the tissue is an innocent bystander.
Precisely. The kidney isn't the target, it's just where the clump got stuck in the filter. But once it's stuck, it does the same thing we saw in type 2. It activates complement and recruits leukocytes. The neutrophils arrive and try to eat the complex, but they can't because it's embedded in the wall, so they release their enzymes in frustration.
And that destroys the vessel wall. Vasculitis.
Yes, fibrinoid necrosis. This is the core mechanism of systemic lupus erythematosus, SLE, and serum sickness. It's collateral damage from a war happening in the bloodstream.
Okay, finally, type 5e, T-cell mediated.
Right, the first three were all about antibodies. Type 5A is different. No antibodies involved at all. This is the T cells taking charge. It's called delayed because T cells take 24 to 48 hours to mobilize and recruit their backup. It's not immediate.
So if I get a TB test, the MAN2 test, why do I have to wait 2 days to see if it worked?
Because that's how long the recruitment takes. They inject a bit of TB protein under your skin. If you've been exposed to TB before, your memory CD4 plus T cells recognize it. But they can't kill it alone. They have to call for backup.
The generals again.
The generals again. They secrete cytokines, like interferon gamma, to recruit and activate macrophages to the site. That accumulation of angry macrophages is what causes that hard red bump, the induration, a day or two later. That is cytokine-mediated inflammation.
And this is also the mechanism in things like poison ivy. Right?
Exactly. Contact dermatitis, a chemical from the plant binds to your skin proteins, your T cells see this new complex is foreign, and you get that itchy, blistering rash a couple days later.
And the other form of type 5e is direct killing.
Direct cytotoxicity. This is the CD8 plus assassins. They're killing cells directly. This plays a huge role in type 1 diabetes, where T cells methodically seek out and kill the insulin-producing beta cells in the pancreas.
So summing up hypersensitivity, type I is allergy and IgE. Type 2 is direct antibody attack on a cell or tissue. Type 3 is immune complex, the clumps getting stuck. And type 4 is T cells and delayed.
That is a perfect scaffold.
So in hypersensitivity, we have a working army that is just trigger happy or aiming at the wrong target, pollen, dust, even our own cells sometimes. But the soldiers are still loyal, they're just confused.
Right. They're trying to protect you, but they're causing massive collateral damage in the process.
But now we have to go darker. What happens when the loyalty itself is broken? When the army looks at the king, the body's own tissues, and decides he is the enemy.
That is the breakdown of tolerance. That is autoimmunity.
Part 3, autoimmunity. This brings us to a core philosophical question of immunology. How does the body know not to attack itself?
Robbins calls this immunologic tolerance, and the answer is surprisingly risky. We don't have a genetic blueprint that says don't attack self. Instead, we produce billions of random lymphocytes that could attack anything, including us.
Wait, hold on. You're saying we possess cells that are specifically designed to kill us and we just hope they get deleted. That seems like a terrible evolutionary design.
It seems that way, but it's high risk, high reward. to recognize any possible invader you might ever encounter, from a virus that doesn't exist yet to a weird fungus. The body generates random receptors through gene rearrangement. By definition, some of those random roles will target your own heart or liver or brain.
So it's the price of admission for having an infinitely adaptable defense system.
Exactly. But we have a boot camp to weed them out, a very brutal boot camp.
This is central tolerance.
Right. It happens in the primary lymphoid organs, in the thymus for T cells and the bone marrow for B cells. As these young cells, these recruits, are maturing, they're presented with a whole library of self-antigens. It's a test.
A loyalty test.
A loyalty test. If a recruit reacts strongly to a self-antigen, if it shoots the drill instructor, it is commanded to undergo apoptosis. It's deleted.
You're too dangerous, you die.
Exactly. Negative selection. But the system isn't perfect. Some self-reactive cells slip through the cracks.
So we have sleeper agents in the blood.
Correct. So we have peripheral tolerance. These are the backup mechanisms out in the tissues. For example, if a self-reactive T cell that escaped the thymus sees its antigen on a normal cell, but doesn't get that second signal or co-stimulation from the innate system.
A danger signal.
Danger signal. It shuts down. It becomes anergic or unresponsive. It's disarmed.
Or there are the regulatory T cells, the TREGs.
Yes, the peacekeepers.
Yeah.
They actively suppress other T cells to prevent them from attacking self. They're like the military police of the immune system.
So autoimmunity happens when all of this fails.
Yeah.
The central deletion, the peripheral energy, the
Yeah.
TREGs. What causes that kind of catastrophic failure?
It's usually a perfect storm. You need two things, a genetic susceptibility and an environmental trigger.
Let's look at the genetics. Robbins emphasizes the HLA alleles.
The HLA genes are by far the strongest dissociation. These are the genes that code for the MHC molecules we talked about, the plates that serve the antigen.
Right.
Certain shapes of plates might be better at picking up and presenting a particular self-antigen to the immune system. For example, if you have the HLA-B27 allele, your risk of ankylosing spondylitis is massively higher than someone without it. The plate is just the right shape to serve up a self-peptide from your joints.
But genes alone aren't enough. You can have the HLA-B27 gene and never get the disease. You need a trigger.
Right. Environmental trigger. Infection is a huge one.
How does an infection like a virus lead to attacking yourself?
One major way is molecular mimicry. A microbe happens to look very similar to a self-antigen. The classic example is rheumatic fever.
Okay.
You get a strep throat. The immune system mounts a vigorous attack against the strep bacteria. But a protein on the surface of the strep bacteria, the M protein, looks just like a protein in your heart valves.
So the immune system, in trying to kill the bacteria, ends up recognizing and attacking the heart.
Exactly. It's a case of mistaken identity. The antibodies and T cells cross-react. Another mechanism is epitope spreading.
This sounds ominous. What is spreading?
The attack. Imagine you have a small, localized autoimmune reaction. Maybe T cells start attacking one specific protein in your skin.
Okay.
That attack causes tissue damage. Cells die and break open. And that releases other hidden self-antigens from inside the cell that the immune system hasn't seen before. The immune system says, hey, here's more foreign stuff, and it mounts a response to those new targets, too.
So the war front expands. The list of targets gets longer.
It does. The target gets bigger and bigger. This explains why many autoimmune diseases are progressive and get worse over time.
Let's look at the prototype of autoimmune disease, systemic lupus erythematosus, or SLE.
The great imitator. It's called that because it can affect any organ system. SLE is a multi-system disease of failed tolerance. It affects the skin, joints, kidneys, blood cells, brain, everything.
And the hallmark here is the presence of autoantibodies, specifically anti-nuclear antibodies, or ANAs.
Yes, antibodies against the stuff inside the nucleus of a cell, against DNA, against histones, against ribonucleoproteins. The really specific ones for lupus are anti-double-stranded DNA and anti-Smith antigen, or anti-SIMA.
Robbins has a fascinating insight into the mechanism here. It's a vicious cycle involving cell death. Can you walk us through the narrative of a lupus flare?
Sure. Imagine a young woman who has the genetic predisposition for lupus. She goes to the beach for the day. UV light from the sun hits her skin.
Okay.
UV radiation damages DNA and causes some of her skin cells to undergo apoptosis, or programmed cell death.
Okay, cell death happens all the time. Our bodies are constantly turning over cells. Why is this a problem?
Normally, macrophages come in and quietly clean up the debris from these dead cells. It's a very tidy process. But in lupus, that clearance mechanism is defective. The dead and dying cells linger for too long. Their nuclei break open.
So you have nuclear trash DNA, histones floating around in the tissue.
Exactly. And because because of her underlying loss of tolerance, her B cells see that nuclear trash as foreign. They think it's a virus. They get help from T cells and start making high-affinity IgG antibodies against it.
And now we are right back to type 3 hypersensitivity.
Exactly. The anti-DNA antibodies bind to the free DNA. You form immune complexes. Those complexes travel through the blood and deposit in the kidneys, causing nephritis, in the skin, causing the classic butterfly rash, and in the joints, causing arthritis.
And then the inflammation from that deposition causes more tissue damage, which releases more nuclear antigen.
And the cycle fuels itself. It's a self-perpetuating loop of injury. That's why the disease has this relapsing and remitting course. A trigger causes a flare, which causes damage, which creates more fuel for the next flare.
It's a tragic mechanism. We should briefly mention a few others in this category. Rheumatoid arthritis.
That is mainly a destructive arthritis of the joints. It's driven by T cells and antibodies, particularly against proteins that have been modified in a process called citrullination, and you get something called rheumatoid factor.
Sjogren's syndrome.
That's the classic dry eye, dry mouth disease. It's a T cell-mediated attack that destroys the lacrimal and salivary glands.
And systemic sclerosis, or scleroderma. That word always sounds scary.
It is. This is where the immune system triggers excessive fibrosis, or scarring. The skin tightens, the esophagus hardens, the lungs become fibrotic. It's driven by vascular injury and T cells pushing fibroblasts to overproduce collagen. The body essentially turns to stone.
It's scary stuff. But now let's flip the coin. What happens when the system doesn't do enough? Part 4, immunodeficiency.
The gap in the shield. The opposite problem, but just as deadly.
Underwood gives like a great clinical clue here. Septurar.
Yes. Severe, persistent, unusual, and recurrent infections. If you see a patient with infections that fit that pattern, you have to think about immunodeficiency.
We divide these into primary and secondary. Primary means you're born with it, right? A genetic defect.
Yes, genetic or inborn errors. These usually show up in infancy, often after about 6 months of age.
Why 6 months? Why not immediately at birth?
Because for the first 6 months, the baby is borrowing the mother's immune system. Maternal IgG antibodies cross the placenta during pregnancy. They provide passive immunity that protects the newborn. But by 6 months, those maternal antibodies degrade. The baby's own system has to take over. If it can't, that's when the infections start.
Let's hit the big ones. Defects in adaptive immunity.
Okay, first, X-linked agammaglobulinemia, also called Bruton's. This is a defect in a gene on the X chromosome called BTK-Bruton tyrosine kinase.
What does that kinase do?
It's an essential signal for B cells to mature. Without it, B cells get stuck in the pre-B stage in the bone marrow. They never make it out into the blood.
So no B cells means no antibodies.
Zero. No plasma cells, no antibodies.
Yeah.
These kids get recurrent bacterial infections, sinusitis, pneumonia, because they can't opsonize bacteria.
Then there's DeGeorge syndrome. This is a developmental issue, right? Not a single gene.
Correct. It's a deletion on chromosome 22 that messes up the development of the third and fourth pharyngeal pouches in the embryo. That leads to no parathyroid glands, so low calcium, and crucially, no thymus, or a very small one.
And the thymus is the school for T cells.
Exactly. No school, no graduates. You have thymic hypoplasia leading to a profound lack of T cells. Without T cells, you can't fight viruses or fungi effectively.
And then the worst case scenario, SCID, severe combined immunodeficiency.
The bubble boy disease. This is where you have defects that knock out both B and T cells. A common cause is a deficiency in an enzyme called ADA adenosine deaminase.
How does one enzyme deficiency wipe out the entire adaptive immune system?
Without ADA, toxic metabolites build up inside the lymphocytes and kill them. It's particularly toxic to the stem cells that give rise to both B and T cells. You have no adaptive immunity at all. It is fatal early in life without a bone marrow transplant.
But Robbins notes that the most common primary immunodeficiency is actually much milder, isolated IgA deficiency.
Yes. By far the most common. Many people are asymptomatic, but some get more mucosal infections, recurrent sinus infections, lung infections, diarrhea, because IgA is the main antibody that protects our mucosal surfaces.
Now, secondary immunodeficiencies, these are acquired and much, much more common.
Absolutely. Malnutrition is probably the biggest cause worldwide. Cancer, chemotherapy, immunosuppressive drugs for autoimmunity or transplants, these all suppress the immune system. But the most significant infectious one is HIV/AIDS.
Let's connect this back to our earlier discussion of the generals. How does HIV work mechanism-wise?
HIV is a retrovirus. It has a protein on its surface called GP120. That protein is a perfect key for the CD4 molecule.
The very molecule that identifies the helper T cells.
Yes, the virus specifically targets the generals. It binds to CD4, gets inside the cell, uses reverse transcriptase to turn its RNA into DNA, and then integrates that DNA into the host cell's genome. It turns the cell into a permanent virus factory. Eventually, the cell dies.
So it's a systematic assassination campaign against the leadership of the immune system.
Exactly. As the CD4 count drops and drops over years, the entire coordination of the immune response collapses. The B cells don't get the signal to make effective antibodies because there are no helper T cells to shout, "class switch." The CD8 cells don't get the signal to attack. The macrophages don't get activated.
And the consequence is AIDS.
Acquired immunodeficiency syndrome. The patient doesn't die from HIV itself. They die from opportunistic infections. Microbes that a healthy immune system would swat away without even blinking. Pneumocystis, pneumonia, esophageal candida, Kaposi sarcoma.
It really highlights how absolutely critical those CD4 cells are. Without them, the army is just a useless, disorganized mob.
Precisely. It's the perfect illustration of why immune regulation and coordination are everything.
We have one brief section left before we conclude. Amyloidosis. This always feels like the odd one out in pathology chapters. A bit strange.
It does, but it's often linked to immunity and inflammation. Amyloidosis isn't a single disease. It's a condition where abnormal proteins misfold. They adopt this beta-pleated sheet conformation and deposit in tissues.
Robin says it's called amyloid because it stains with iodine and looks starch-like.
Right, and these insoluble protein deposits physically compress and damage organs, leading to things like kidney failure or restrictive cardiomyopathy.
And there are two main forms relevant here.
Yes. First is AL amyloidosis. The A is for amyloid and the L is for light chains.
Light chains? Those are parts of antibodies.
Exactly. This is often associated with multiple myeloma, a cancer of plasma cells. The cancerous plasma cells churn out huge amounts of a single type of light chain, which then misfolds and deposits all over the body.
So it's a disease of too much antibody protein getting stuck.
Basically. And then there's AA amyloidosis, which is reactive. The AA stands for amyloid associated. This is associated with chronic inflammatory states like rheumatoid arthritis or inflammatory bowel disease.
How does that work?
During chronic inflammation, the liver pumps out a protein called serum amyloid A. It's an acute phase reactant. If the inflammation goes on for years and years, eventually that protein starts to misfold and deposit in the kidneys or heart.
So it's a long-term consequence of the immune system running hot for too long.
Exactly. It's the debris of a never-ending war clogging up the machinery of the body.
This has been a massive tour. Let's try to bring it all together.
Okay. We started with the architecture, the elegant balance of innate barriers and adaptive specialists, the guards who see general patterns, and the snipers who see specifics.
Then we saw what happens when the reaction is excessive hypersensitivity, from the explosive, orchestrated allergy of type 1 to the plumbing problems of type 3 complexes getting stuck everywhere.
Then we saw the tragedy of autoimmunity, where the fundamental principle of self-tolerance fails, and the body turns its most powerful weapons on itself, like in the vicious cycle of lupus.
And finally, we saw the vulnerability of immunodeficiency, where the shield is missing gaps, either because of a genetic defect or because a virus like HIV specifically targets and eliminates the leadership.
So what, here for you, for the listener, is that immune pathology is dynamic. It's a balance. It waxes and wanes. It's not just you have a disease. It's your regulation is failing.
And understanding the mechanism dictates the treatment. If it's an antibody problem, you treat it differently than a T-cell problem.
Exactly. You don't treat myasthenia gravis, which is a type 2 antibody-mediated disease, the same way you treat contact dermatitis, a type 5e T-cell-mediated disease. The biology dictates the therapy.
I want to leave everyone with a final provocative thought that comes up in Robbins, the hygiene hypothesis.
Ah, yes. It's the idea that in our modern, ultra-clean world, with widespread sanitation and decreasing childhood infections, our immune systems are getting, well, bored. Or maybe undertrained. Think about the Th2 system. It evolved over millennia to fight parasites, to fight worms. If we eliminate parasites from our environment, what does that powerful pre-programmed system do with its time?
It looks for other targets.
The theory is that without enough real threats to fight, the system turns its attention to harmless things like pollen leading to allergy or even self-antigens, contributing to autoimmunity.
So, are we seeing more of these diseases because we are too clean? Because we've removed the things that used to keep our immune systems properly calibrated?
It's a fascinating and controversial area of research. It suggests that a little bit of dirt, a few infections in childhood, might be necessary to teach the immune system what not to panic about, to properly educate it.
Something to mull over. As always, this deep dive is just the map. The territory is in the books. Open Robbins, open Underwood, look at the beautiful diagrams of the hypersensitivity types, read the section on SLE pathogenesis.
The nuances in the text, the details are there. Use this as your framework and go find them.
Thanks for listening to the deep dive. We'll see you next time.